Hello and welcome to this BioPharma Dealmakers webcast on new innovations in drug delivery technology. My name is Ravina Bambra and I am the editor of BioPharma Dealmakers. I will be your moderator today. The demand for innovative drug delivery technologies is increasing, particularly due to the continuing commercial success of biologics such as monoclonal antibodies, as well as growing interest in cell and gene therapies, for which delivery can be a key challenge. The diverse drug delivery market is therefore now seeing major developments in digital devices and implantables, as well as transdermal, fibre-based, nano and matrix technologies to meet this growing demand and provide targeted medications in therapeutic areas such as oncology, CNS diseases and women's health. In this webcast, we will hear from 5 speakers from the drug delivery field who will each present the technologies their companies are developing. After the presentations, we will have a roundtable discussion with the speakers and a question and answer session to allow you to participate and contribute your own thoughts. To ask the speakers a question, just press the Ask a Question button at any point during the presentation and we will answer them. Our first speaker today is Dr. Patrick Anquetil, Chief Executive Officer of Portal Instruments.
Good morning. My name is Patrick Anquetil and I'm the CEO and co-founder of Portal Instruments. Portal Instruments is a novel needle-free drug delivery system aiming at solving an overlooked challenge, which is the administration of viscous biologics for patients suffering from chronic diseases. Biologics, as you know, need to be injected, and that leads to a subpar patient experience. We would like to transform this experience with a novel technology issued from MIT, and that is based around the controlled jet technology. The invention and the seminal patent portfolio comes from Ian Hunter's group at MIT, who is a professor in mechanical engineering. Our business model is to partner with pharmaceutical companies to help them better deliver their drugs to patients. To date, we've done extensive preclinical and clinical work and are transitioning to the commercialization phase. At the bottom of the slide you can see our partners. Sanofi and 3M are our first investors and were the first corporate believers in our technology. We've also added 5M Ventures, who led our Series B, and in aggregate we've raised $36 million of financing. Our team is composed of experienced executives in engineering, manufacturing, quality, regulatory affairs and software development. And as you can see, we have an illustrious scientific advisory board. The challenge with biologics is that today there is no other way but to inject them. In addition, with the advent of self-injection devices, the burden of the administration has been transferred to the patient. And that creates a lot of anxiety. And this anxiety is not necessarily phobia. It starts with an emotion. And that emotion is that the patient feels anxious about taking this injection because if he or she doesn't get it right, they may not get well, they may not get better. No one likes needle, and I think this is something quite universal across humans. In particular, if you inject a biologic, you tend to experience a slow and painful injection because of the viscosity and the larger volume of the drug. In fact, in some cases, the patient needs to be augmented with an auto-injector, as seen on the bottom right of the slide, because he or she may not have enough force in their thumb to perform the injection because of the high viscosity of that drug. Safety concerns are very real. If you have young children, for example, you always worry about having needles around. Patients have also told us that needle disposal is an issue, as very few places take them anymore. Finally, the cost per injection is also a challenge that we hear from our pharmaceutical partners. In fact, auto-injectors, as you know, are disposable devices, and those have a non-trivial fixed cost. What if we could transform this experience and have the therapy fit into the patient's life? What if he or she got, first of all, a reminder when it's the right time to take their medicine? The patient could then grab a single-use disposable cartridge that comes pre-filled by the pharmaceutical company. Then the patient would load the cartridge onto the device, perform the administration, and dispose of the cartridge in the regular trash, since there are no sharps. We've then also augmented the therapy experience with cloud-based services. Once injection is completed, the device transmits automatically the injection data into the cloud. The data can be read by an app or web browser and provides support, motivation, and even education on the disease. For example, on this slide, you can see the app that allows the patient to know when is the next dose, how well doing the therapy, and even gives them an ability to record symptoms. This data in turn can be shown to the physician at the time of the visit and empowers the patient to have a data-driven dialogue with his or her physician. This slide shows the cloud-based ecosystem that we've built. The ecosystem connects all stakeholders to the therapy with real-time data from the physician, caregivers, pharmacies, but also the pharma companies and the payer. This platform is available today for our partners, completely independently of the device, and we look forward to partnering it up with you. So what is our value proposition? Well, our value proposition is centered around three key themes. Number one, by removing the needle, we transform the patient experience, which leads to a de facto differentiation versus other therapy and potentially a market share gain. Number two, we are also able to enhance market penetration because we can reach patients who refuse a therapy because it is needle-based if they have an alternative. And as you know, sometimes the alternative can be do nothing. Number three, finally, because of the transformed nature of the experience and the enhancement of the therapy with our digital health cloud, we think we will be able to also have an impact on patient adherence, which is, as you know, another challenge. So what is a portal solution? At the core of it is a powerful computer-controlled electromechanical drive mechanism that produces a precise and fast jet. We can inject a 1 ml solution in a third of a second. We are completely agnostic to drug viscosity up to 200 centipoise, and I'll get into this very shortly. As you've seen in the previous slide, the container can also be disposed in a regular trash since it has no sharps. The way the portal device works is that we create a jet using an electromagnetic actuator on a closed-loop feedback computer control. This is equivalent to what you have in your car when you place it in cruise control or auto cruise. There is a computer system in the car that makes sure that the right amount of velocity is maintained irrespective of terrain. For example, it will slow down the car if you go down a hill. Very similarly, the portal device has a computer system that monitors how much drug has been injected and corrects the speed of the injection if it detects that it has not injected the right amount of drug. What this means is that we can place a non-viscous solution or a very viscous solution, say a 200 centipoise solution, and without changing anything, the device will automatically compensate for that increased viscosity and perform the same 1 ml solution injection in a third of a second. As you can see in this slide, the performance of the device does not get degraded as we increase the viscosity of the solution. In this slide we show how we've studied the sensation that the subject feels when he or she is injected with our device and compares it to a 27 needle and syringe injection. In such head-to-head comparison, we can demonstrate that our device is less painful than a typical needle and syringe. In fact, using a visual analog scale where 0 is no pain and 100 is the highest pain ever experienced, we can show on average a 40% reduction in sensation. Following those injections, we then asked the subjects about their experience and which device they preferred. We are excited to report that in 78% of the cases, they preferred our device versus only 14% of the case for the needle and syringe. And this establishes a great preference for our device. We've also spent a great amount of time studying the question whether our device could affect a drug because of the high pressures it produces. To date, and together with 8 pharmaceutical partners, we've shown across 16 biologics that our device has no impact on a drug's physical integrity or on the functional integrity of that drug. Finally, we've also done 2 PK studies in animal models, and we've shown a close to 100% equivalence to a needle and syringe. Finally, I'd like to highlight that we're ready for prime time. We are ISO 13485 certified as of a few months ago, and we've also developed a primary drug container that is compatible with typical biologics. So all you have to do is take your container, put your drugs in, and start your studies. In summary, Portal Instruments has developed a transformative technology solving a century-old challenge which is a patient experience during the administration of injectable medicines. The challenge is particularly acute for patients needing to self-inject because of chronic conditions. We have a unique needle-free jet injection technology that is augmented by cloud-based services. We've done a tremendous amount of preclinical and clinical work to support our value proposition. In fact, we've shown that our device, in a head-to-head comparison with an injection with a 27-gauge needle and syringe, is preferred 78% of the time. We're moving at this time into commercial partnerships with our different pharmaceutical partners, and we look forward to having more conversations with other potential partners and together to solve your challenges in drug delivery. Thank you very much for your attention. I look forward to a great discussion during the Q&A part of this session.
Thanks, Patrick. Our next speaker is Dr. Joseph Sarrett, Chief Business Officer of Corium International.
Thank you. It's a pleasure to have the opportunity to speak with you today. Corium is a commercial stage biotechnology company dedicated to expanding the reach of transdermal products. In addition to our technology platforms and research capabilities, we also have a commercial-scale transdermal manufacturing facility. As a result, all of our development work is done with an eye to ensuring that the process is scalable and economic. We have a pipeline of products in the CNS space, and our lead product is a once-weekly transdermal formulation of donepezil, the most commonly prescribed agent for Alzheimer's and the active ingredient in Aricept. We have two main technology platforms. The first is Corplex, which is a passive system for the transdermal delivery of small molecules. This system is best suited for chronic therapies, where having sustained and controlled blood levels of the therapeutic agent is important. Microcore, which is a dissolving microneedle technology, is designed for the active delivery of potent large molecules, including vaccines. It is an integrated, pain-free system without the refrigeration requirements typically associated with large molecule storage and delivery. In addition to developing our own proprietary pipeline, we work with partners to incorporate their APIs into either of our platforms. Today, we'll be focusing on our Corplex technology platform. The power of the Corplex platform is related to the blending of both hydrophobic and hydrophilic polymers within the same membrane. The hydrophobic elements provide excellent adhesive properties. Our goal is to develop multi-day, ideally once-weekly patches that adhere during normal daily activities, including bathing and exercise. The hydrophilic elements allow the patch to handle the moisture in the skin and allow us to achieve the high drug loading necessary to maintain consistent flux across the skin over the course of 7 days. The patches are very thin and flexible, resulting in an improved patient experience and comfort. We also have a robust IP portfolio covering both the technology platform and the unique formulations that result from the incorporation of specific APIs into the Corplex system. This is an example of how the Corplex technology can deliver improved flux across the skin. The graph on the left shows that we were able to achieve equivalent plasma concentrations of lidocaine as those observed with the commercial lidoderm patch. But we were able to do this with only 20% of the drug loading used in lidoderm. In other words, we were 5 times more efficient in delivering lidocaine than the commercial product. Not only does this help reduce costs, it also minimizes the amount of drug left in patches after use, which is an increasing focus of the FDA and other regulatory agencies. Even more importantly, for drugs that are particularly difficult to get across the skin, the ability of Corplex to enhance flux can create first-in-patch opportunities. We are focusing our internal development efforts on using our Corplex technology platform to develop a pipeline in the CNS space, where we think the rationale for transdermal delivery is particularly compelling. For chronic diseases like Alzheimer's and Parkinson's, having consistent, sustained plasma concentrations of therapeutic agents is important, and transdermal delivery can significantly improve compliance, particularly in patients that have swallowing difficulties, which are common in this patient population. Another important benefit of transdermal delivery is the potential to significantly improve the adverse event profile. By avoiding the GI tract, we can reduce local GI side effects and can avoid the first pass effect. In addition, we can reduce or eliminate adverse events related to either Cmax or the rate of rise in plasma concentrations. Our 2 lead products are donepezil and memantine, the 2 most widely prescribed agents for Alzheimer's disease. In each case, we are developing once-weekly formulations of the active agent. As you can see here, donepezil, shown in blue, is the most common drug at all stages of disease. Memantine is typically added as the disease progresses, and the two agents are frequently used in combination. This slide shows the results from our 1-week phase 1 study of Corplex donepezil. The orange lines show plasma concentrations of donepezil from oral Aricept on day 1 and day 7. As you can see, plasma concentrations on day 7 are significantly higher than on day 1. This is due to the long half-life of donepezil, which is approximately 70 hours. The solid blue lines show the plasma concentrations over the week from a single application of the Corplex donepezil patch. Again, plasma concentrations increase over the course of the week, though the transdermal formulation results in a more gradual and sustained increase in plasma concentrations compared to oral Aricept. It is worth noting that it is particularly difficult to get consistent delivery of donepezil across the skin, and this is one of those first-in-patch opportunities that I mentioned earlier. Based on our phase 1 data, we proposed a bioequivalence, or BE, approach to approval to the FDA. This approach is based on a simple PK comparison of plasma levels of the drug administered via the approved daily oral dosage form versus the once-weekly patch in healthy volunteers. After reviewing our phase 1 data, the FDA agreed with this approach. Key to this determination is the long half-life of donepezil, and the PK comparison will be made at steady state, which occurs after 3 to 4 weeks of therapy. For comparison, without the BE approach, a full efficacy study in Alzheimer's patients would be required, so the savings from the BE pathway in terms of time, cost, and complexity are substantial. In a separate meeting with FDA regarding our once-weekly memantine program, the agency again agreed that the BE pathway is acceptable, with no requirement for efficacy testing. We have also received preliminary guidance from a major European health agency suggesting that the BE pathway would likely be acceptable there as well. In November of last year, we started a pilot bioequivalence study for Corplex donepezil. This study is a 3-way crossover study in healthy subjects, with 5 weeks of dosing for each treatment period and a 3-week washout interval between periods. In addition to oral Aricept, we are testing 2 versions of the patch, which each contain the same formulation and differ only with respect to patch size. We measure PK results during the 4th week of the 10 mg dose, when plasma levels are at steady state. At the end of the study, each subject will have received all 3 treatments. The next slide shows our projections of what plasma concentration should look like at steady state based on the 1-week phase 1 data we discussed earlier. Again, oral Aricept projections are shown in orange, and projections from the transdermal patch are in blue. To demonstrate bioequivalence, we must show that the area under the curve, or AUC, and Cmax from the transdermal application are within 80 to 125% of the values seen with oral administration. AUC is measured over the course of the entire week. Slide 12 shows the interim results from period 1 of our pilot BE study. As in the prior slide, results from oral Aricept are shown in orange. The solid blue line shows the plasma concentrations from the smaller of the 2 patches, while the gray line shows the results using the larger patch. These results are exactly what we had expected to see. While we will need to analyze the crossover data to make a final determination, these data suggest that the ultimate patch size will be somewhere between the 2 patches that were tested. This data gives us a high degree of confidence that we will be able to achieve statistical bioequivalence in our pivotal study. Our preliminary data also suggests that transdermal delivery of donepezil could result in a lower rate of GI side effects. During period 1 of the pilot BE study, 50% of the subjects on oral Aricept experienced nausea, compared to only 25% of the subjects on the larger of the patches, and none of the subjects wearing the smaller patch. Rates of vomiting, while lower, showed a similar trend. We also had excellent skin tolerability, with rates of skin irritation less than seen with many commercial transdermal products. Here we show the timelines to commercialization. As I mentioned earlier, we are in the midst of our pilot BE study, with top-line results from the full study expected in the May-June timeframe. We plan to start our pivotal study in the second half of this year, with the results available early next year. This will enable us to file our NDA in the second half of 2018, with approval in the second half of 2019. Memantine is following a similar plan, but is about 12 to 18 months behind donepezil. As we've discussed, Corplex provides several important advantages. We can expand the number of drugs that can be delivered transdermally, and administration by this route leads to more sustained and controlled drug plasma levels. We typically aim for a once-weekly application, which further improves compliance. And Corplex has excellent skin adhesion with minimal skin irritation. In addition, we can significantly improve the adverse event profile. Finally, we can provide important competitive advantages through blocking IP, surrounding both the technology platform and the specific combination of the platform with each individual API. We are currently seeking global or regional partners for our Corplex donepezil and Corplex memantine product candidates. In addition, we work with partners to develop novel transdermal products by incorporating their API into the Corplex system, providing many of the same advantages we just discussed. We also have the ability to serve as a manufacturing partner for transdermal products that have already been developed. In sum, we have a long track record of successfully developing and manufacturing transdermal products for partners, and we are now leveraging that expertise to use our proprietary platforms to develop our own pipeline focused on the CNS space. In addition to our once-weekly formulation of donepezil, we have a pipeline of other transdermal products focused on the CNS space, including once-weekly memantine, also for Alzheimer's. We are committed to expanding the reach of transdermal products so that patients and their families can enjoy the important benefits that the Corplex system has to offer. Thank you very much.
Thanks, Joseph. Our next speaker is Dr. Kevin Nelson, founder and Chief Scientific Officer of TissueGen.
Thank you for the opportunity to speak with you today. I'm Dr. Kevin Nelson, founder and Chief Scientific Officer at TissueGen. TissueGen was founded in 2000 and has developed and manufactures elute fiber, a biodegradable fiber for controlled, sustained delivery of the broadest range of sensitive biologics and pharmaceuticals. Elute fiber is an ideal material for both implantable and topical drug delivery, and importantly, may serve as an active scaffold for tissue engineering and regenerative medicine applications. Elute fiber simultaneously provides strong mechanical support while also delivering biologics or pharmaceuticals in controlled dosages over sustained release profiles directly at the topical or surgical site. Typically, medical fibers are manufactured through a melt extrusion process at high temperature and high shear stress, severely limiting the types of pharmaceuticals or biologics that could remain viable throughout the manufacturing process. To address these limitations, we developed and patented a modified wet extrusion process to manufacture elute fiber at or below body temperature and with relatively low shear stress. When coupled with other proprietary methods, the pharmaceutical or biologic is protected throughout the manufacturing process, thereby enabling fiber-based delivery of the broadest range of biologics and pharmaceuticals ever possible. Elute fiber can deliver a broad range of sensitive biologics, including, but not limited to, growth factors, immune proteins, enzymes, oligonucleotides, even whole viruses. When used as a three-dimensional scaffold, elute fiber can create positionally stable chemical or biological concentration gradients along specific pathways. This is possible by either creating a concentration gradient down the length of a single fiber or by creating multiple fibers with uniform drug distribution and variable distance within the scaffold. In fact, elute fiber loaded with chemotactic factors for specific cell types can be used to create concentration gradients that encourage the target cells to migrate along pathways that are precisely determined by the fiber and effectively orchestrate parts of the wound healing process. One present application is peripheral nerve regeneration. In long gap injuries, the natural chemotactic factors that entice nerve regeneration are unable to diffuse across the gap, and functional regeneration does not occur. However, an implanted elute fiber scaffold can create stable concentration gradients of these same neurotrophic factors and encourage axonal migration across the gap. This application for elute fiber is currently being tested in a rabbit peripheral nerve model to regenerate perennial nerves across a 40 millimeter gap. Elute fiber can also simultaneously create multiple concentration gradients, targeting different cell types within the same scaffold. TissueGen is currently testing this unique approach to induce spinal cord regeneration. Within the elute fiber scaffold, one set of neurotrophic factors may encourage motor neurons to traverse a gap in the spinal cord in one direction, while a different set of neurotrophic factors encourage sensory axons to traverse the gap in the opposite direction. This approach may promote spinal cord regeneration without the aid of tissue grafts or stem cell treatment. Unlike traditional drug delivery formats, such as microspheres and nanoparticles, elute fiber provides both mechanical and pharmacological support from the same device. Elute fiber is well suited for a wide range of current medical applications, including braids, meshes, and weaves for medical textile applications, monofilament sutures and ligatures, such as those commonly found in orthopedic applications. Also, when elute fiber is manufactured in a non-circular cross-section format, it can provide significant radial strength to serve as a stent. For example, TissueGen has leveraged elute fiber technology to produce the Archer stent, our patented self-expanding medical device offering drug delivery from an implantable biodegradable stent. We specifically designed Archer stent as an expandable helical coil to meet the mechanically challenging environment of the peripheral circulatory system, including the femoral and popliteal arteries of the leg. However, Archer stent is ideal for a wide range of applications where positional stability and radial stiffness are critical during early recovery, followed by gradual degradation of the device in order to return mechanical loading to the healing tissue. As mentioned earlier, we employ several proprietary methods in addition to our patented extrusion process to manufacture elute fiber. While the solvents and non-solvents used in our wet extrusion process enable elute fiber formation at or below body temperature, these same chemicals may destroy the incorporated biologics and pharmaceuticals. Therefore, we developed a method to isolate the biologic or pharmaceutical from the harsh solvent system to protect the drug throughout manufacturing. Following extrusion and prior to use in medical applications, however, the residual solvents must be removed from elute fiber. Therefore, we also developed a post-processing method to decrease residual solvent content to levels well below the allowable limit set by the U.S. Food and Drug Administration. Importantly, these post-processing methods do not denature the biologics or pharmaceuticals loaded in the elute fiber, and the drug retains its viability post-manufacturing. Elute fiber can deliver drugs that range in size from small pharmaceuticals to viruses in controlled, sustained profiles over days, weeks, even a year, based on the drug and fiber properties. The two graphs shown here plot ELISA-detectable cumulative release over time of glial-derived neurotrophic factor, or GDNF, and vascular endothelial growth factor, or VEGF. Both of these growth factors have half-lives on the order of hours to days. However, when delivered from elute fiber, these same growth factors may be delivered sustainably over many weeks. And note the unique profiles achievable with elute fiber. As shown, the GDNF release profile starts with a burst on the order of about 10% of total release, whereas the VEGF-loaded fiber has been tailored to deliver approximately 60% of its total release over a similar time period. We can design elute fiber to provide tailored release profiles by modifying a number of parameters, including fiber size and shape, drug concentration, polymer type and molecular weight, and the degree of crystallinity imparted to the fiber throughout the manufacturing process. We typically manufacture elute fiber from industry-standard biodegradable polymers commonly used in medical devices today. However, we can also work with proprietary polymers specified by our customers. Several polymers that we've extruded include those listed here. TissueGen offers elute fiber in a wide range of physical formats, including monofilament, hollow, bicomponent, core sheath type structures, and even gel-filled center. We tailor the dimensions of elute fiber for each application, including round, flat, rectangular, or ribbon-shaped designs. Because elute fiber is sufficiently strong to be woven, knitted, or braided, it's ideal for medical textile applications that may benefit from localized, non-systemic drug delivery. At TissueGen, we're committed to implementing quality manufacturing practices and supporting regulatory approval for our customers' elute fiber-based medical devices. In 2015, TissueGen received ISO 13485 certification, and we continue to make significant investment in our quality management system, as well as research, development, and production infrastructure. For example, we now manufacture elute fiber in an ISO Class 7 cleanroom facility to support our customers' compliance with FDA CGMP requirements for manufacturing pharmaceutical product. Following the initial release of elute fiber in 2013, we've been very fortunate to receive industry recognition for this unique drug delivery technology, including the 2015 R&D 100 Award in the Market Disruptor Service category and the ITMA Future Materials Award for Most Innovative Small Company in that same year. Today we work with leading pharmaceutical, medical device, and therapeutic companies to develop elute fiber-based products that may deliver the future of medicine. Thank you once again for the opportunity to speak with you today. It's been my pleasure to discuss our work at TissueGen and to share with you what's now possible with elute fiber. Thank you.
Thanks, Kevin. Our next speaker is Dr. Paul Gavin, Chief Scientific Officer of Phosphogenics.
Hello everyone. My name is Dr. Paul Gavin and I'm the Chief Scientific Officer of Phosphogenics. Phosphogenics is a Melbourne-based biotechnology company that has developed a proprietary platform technology called TPM. TPM allows us to overcome a range of drug delivery issues that are currently present in oral, transdermal and parenteral dosage forms, and we apply this technology to drugs or nutrients to increase their absorption, their safety or efficacy. Today I'm going to give you a brief overview on the TPM technology before presenting some specific examples of how it can benefit oral, injectable and transdermal dosage forms. We are a listed company in Australia, so please see our safe harbour statement. So what is TPM? TPM is a mixture of two different phosphorylated forms of vitamin E, a mono and a ditocopherol phosphate, which you can see up on the right. We manufacture TPM in Melbourne from alpha tocopherol using a proprietary phosphorylation process. Tocopherol itself is a chemically unstable oil with poor aqueous solubility, while TPM is a waxy solid with very good stability and increased solubility. Our early work characterized TPM as a novel form of vitamin E, and we demonstrated that tocopherol phosphates have enhanced vitamin E activity for many of the non-antioxidant roles. Now, the addition of the phosphate group also makes TPM more amphipathic. It has a lipophilic carbon tail, but also a phosphate head group able to interact with water. Consequently, when TPM is dissolved in an appropriate co-solvent and added to water, it will self-assemble into vesicles or nanoparticles. The image on the bottom left shows a very simple TPM nanoparticle suspension. In the middle you can see a high magnification view of the nanoparticles, and on the right you can begin to see the shape and internal structure of an individual particle. Generally these particles are highly elastic and can be 100 to 150 nanometers in diameter. The TPM nanoparticles are able to encapsulate molecules inside their core. Now this is especially true for poorly soluble molecules and enables them to be dissolved in aqueous media. You can see a visual example of this encapsulation here. So what you're looking at on the left is vitamin D added to milk. So vitamin D remains as a bright yellow insoluble oil slick on the surface, even though milk already contains a lot of fats and proteins that help keep lipids in suspension. On the right you can see the same formulation with a bit of TPM added. You can clearly see there's no longer insoluble vitamin D on the surface, as the TPM has solubilized the lipid and maintains it within the nanoparticle. This TPM encapsulation can be used for a number of applications across multiple routes of administration. Over the next few slides I'll present specific examples of demonstrating how TPM can be applied to drug delivery problems in injectable, oral and transdermal dosage forms, and how we are using these attributes to develop commercially differentiated products. Let's start with injections. In an ideal world, injections would all be clear solutions in predominantly aqueous vehicles. However, this is not always possible for drugs that have poor water solubility or are readily hydrolyzed. These drugs present a range of formulation challenges, some of which are described on this slide, and the solutions to those challenges are not always ideal. Poorly soluble drugs require the addition of solubilizing excipients and or the removal of water. Some of these excipients, like chromophore, are known to be toxic. Other excipients, surfactants or lipids, can cause allergic reactions or pain upon injection. We've begun investigating whether TPM could be used to dissolve injectable drugs with poor aqueous solubility, replacing nasty co-solvents with a form of vitamin E. The first injectable drug we examined was propofol. Now, the commercial propofol formulation is a blockbuster product used for initiating and maintaining sedation during surgery. Now, despite its success, it has a range of formulation limitations. The drug itself is an insoluble oil that requires the addition of lipids to help form an opaque white emulsion that you can see on the left. These lipids include lecithin, derived from egg products, which can be a trigger for anaphylaxis to individuals allergic to egg, as well as a source of nutrients to support microbial growth if the formulation is accidentally contaminated. Now, we use TPM to help reformulate propofol. The results are presented in the image on the right. Our formulation is clear, which is a key safety benefit. It contains no egg products or anything else potentially allergenic and has no lipid to support microbial growth. This was our first success with TPM in injectables, and we're currently working on a number of other products. Let's now turn to oral dosage forms. For oral delivery, one of the primary challenges can be low bioavailability, which typically affects lipids and poorly soluble molecules. Efficient emulsification in the gut can aid absorption, which is why these molecules are often directed to be taken with food. Now, we hypothesized that TPM's ability to vesicularize and encapsulate poorly soluble molecules may fast-track this absorption process. Now, in an in vitro gastric model, which is represented by the graph on the left, TPM significantly increased the solubility of a lipid-soluble molecule, which was coenzyme Q10, when compared to coenzyme Q10 alone in a vehicle. No increase in solubility was observed using tocopherol or tocopherol acetate. Now these same formulations were tested in vivo to see whether this increase in gastric solubility led to an increase in oral bioavailability. Rats were orally gavaged with the formulations and the blood collected for CoQ10 analysis. The bioavailability of CoQ10 was significantly increased by TPM when compared to CoQ alone or in combination with the other tocopherol derivatives. So how are we trying to exploit this commercially? Well, with the growing population, new strategies are being adopted to increase feed efficiency. That is, the amount of weight livestock will put on per kilo of food consumed. Our Animal Health division is currently examining whether TPM can increase the absorption of feed nutrients so that a measurable performance in weight gain is observed. So far, including TPM in stock feeds has been shown to increase the weight gain of 2 different livestock species. On the graph on the left, you can see the average daily weight gain of weaner pigs, which are up to 14 days old when fed a range of test diets. The control base diet in blue represents the normal commercial feed. The orange group contains a high dose of tocopherol acetate, and the last group contains TPM in grey. The TPM group increased average daily weight gain by 11% compared to the control diet. In an industry where every cent counts, a 3% increase in weight gain is considered commercially significant. Likewise, a study was conducted in broiler chickens across their full life cycle, which is the graph on the right. Again, feed containing TPM was found to increase average daily weight gain of chickens compared to tocopherol acetate and the control feed containing no TPM. This increase versus the control was 5.7%. These studies are ongoing, but it seems fairly clear that TPM can add value to oral feeds. The last route of administration we will discuss is transdermal. Now it's probably the hardest route of administration to tackle simply because skin is such an effective barrier. For this discussion, I'm going to jump straight into an example, which is our transdermal oxymorphone patch. Now, opioids represent a mainstay of therapy for chronic pain patients, and when used correctly, opioids are both safe and efficacious. However, opioids come with a range of adverse side effects. Now, when you digest an oral opioid, as represented by the red line in this graph, blood concentrations rise rapidly, crossing the minimum therapeutic plasma concentration to provide pain relief. However, the rapid rise and high Cmax values are associated with adverse side effects, as well as euphoria, which is the incentive for abuse. The troughs in blood concentration between pills result in breakthrough pain, which is the major impediment to good quality of life for these patients. In theory, transdermal delivery offers much for the treatment of chronic pain, as it circumvents peaks and troughs in blood concentration that result from oral absorption and frees elderly patients from taking multiple pills per day. On paper, oxymorphone looks ideally suited for transdermal delivery. It has good potency relative to the other opioids on market, it has a relatively long half-life, and it has poor oral bioavailability. However, oxymorphone doesn't have suitable chemistry to pass through the skin on its own at amounts that would be considered therapeutic. We've spent considerable time developing a TPM oxymorphone patch that overcomes this delivery problem. Phosphogenics has completed several phase 1 clinical trials profiling the delivery and safety characteristics of this oxymorphone patch. Subjects received 3-day applications of the patch and blood analysed for drug content during this period. What we found from these studies is that the patch could deliver at a linear sustained rate for the entire 3 days of application. You can see that the PK profile is very similar to the idealised delivery profile shown on the previous slide. All patients received therapeutic plasma concentrations of oxymorphone. Now we know these concentrations are therapeutic because they are well above the Cmax values reported for the lower strengths of oral oxymorphone, which is currently on market. Overall, the oxymorphone delivery profile looks ideally suited to manage chronic pain around the clock with minimal episodes of breakthrough pain. So from our perspective, these trials have proven that we have a therapeutic product. To conclude, I just thought I'd share a sample of our pipeline. Now, whilst this is not an exhaustive list of everything that we're up to, this neatly summarises the wide variety of drugs, dosage forms and routes of administration that TPM can add value to, by overcoming drug delivery issues related to the parent molecule. A number of these programs already have partners on the strength of the data generated to date. Phosphogenics continues to examine further products across both human and animal health with delivery issues that TPM could potentially help. Now, the 10-minute time frame here was obviously only sufficient to scratch the surface of what we've done with TPM, so please, if anyone has any further questions, feel free to reach out and we'll do our best to answer. Thanks for listening.
Thanks, Paul. Our final speaker for today is Dr. Roy Fafara, Chief Scientific Officer at Triox Nano.
Hello, my name is Dr. Roy Farfara. I'm the CSO of Triox Nano. I would like to thank you for the opportunity to present Triax Nano and its breakthrough nanotechnology platform called SMART. Please notice this slideshow includes more information than is narrated, so feel free to revert to it and go over the more in-depth information found inside later on. Please spare a few moments to read the disclaimer. Triax Nano is an Israeli innovative biotechnology company with the goal of commercializing its patented SMART nanotech drug delivery platform for the effective treatment of human disease. Our first objective is treating triple-negative breast cancer. We have been able to show up to 20 times better ability to concentrate chemotherapy at the target cells. SMART, acronym for Stimuli Multi-Adjusted Responsive Nanotechnology, is an exciting and novel nanotechnology platform created by the synergistic combination of two major nanotech disciplines. We combine between mesoporous nanoparticles and DNA molecular machines. Mesoporous nanoparticles are particles that have many pores. They are constructed from silica with a diameter of 100 to 300 nanometers. Each mesoporous nanoparticle incorporates hundreds of 3 nanometer diameter pores. These pores are filled with a wide range of APIs, active pharmaceutical ingredients. DNA molecular machines are single-strand non-helix DNA molecules, recently invented and not found in the nature. These molecular machines have the ability to sense and respond to their surrounding. For example, a DNA enzyme, which is depicted here, is a DNA machine that has the ability to catalyze other nucleotide chains dependent on elements such as ATP and magnesium. When these elements are abundant, they lock into and change the DNA enzyme's spheric structure, thus activating it. These DNA molecular machines are placed on top of each of the pores and act as intelligent gatekeepers, allowing the intelligent release of the API that is loaded in advance into these pores. The gating system's intelligence is attributed to the specific pre-programmed characteristic of the DNA molecular machine on top of the pores. SMART, standing for Stimuli Multi-Adjusted Responsive Technology Platform, opens a window to a vast field of inventions, comprising intelligent nanoscale computerized machines that are able to sense a combination of multiple variables, at least 2 in their environment, and exert action of unloading active payloads based on pre-programmed mechanism. We start by creating the mesoporous nanoparticle. Mesoporous means the particle has many pores. The total particle diameter is 100 to 300 nanometers, and silica was chosen to construct the particle skeleton. Silica was chosen due to its inert and biocompatibility characteristics. We control many parameters of the particle, such as the particle diameter, pore depth, number of pores, and the total surface area. The silica is then loaded with the active pharmaceutical ingredient, in this case doxorubicin. The loaded API, which is seen in the blue stars, is capped by the SMART caps, for example a DNA enzyme cap, which is depicted in the purple-red structure. The DNA enzyme responds to threshold levels of ATP, cell's primary energy unit, and the ion magnesium, by cutting itself free from the pores and in turn allowing the release of doxorubicin. The number and type of variables that could potentially be incorporated into SMART sensing mechanism are vast. Triax Nano has discovered tens of SMART particles incorporating different intelligent gating systems such as DNA enzymes, aptamers, and enzyme-sensible caps, loaded with different APIs which could be chemotherapies, immune-modulating drugs, siRNAs, or isotopes. SMART platform opens a window to a vast field of innovation. SMART particles are intelligent nanoscale biologic computerized machines able to sense a combination of many variables and/or XOR in their environment, and respond exerting different actions such as unloading different APIs based on pre-programmed mechanism. The number and types of variables that could potentially be incorporated into SMART sensing mechanisms are vast. Triax Nano has discovered tens of SMART particles incorporating different intelligent gating systems. DNA enzyme, aptamers, and enzyme-sensible SMART caps are able to respond to different variables. In vitro efficacy was proven with the following sensing variables: ATP, the cell's energy level, metabolites level, such as lactic acids, enzyme levels, found in higher levels in cancer cells compared to normal cells, and metal ion concentrations such as magnesium and zinc. Again, these variables are required to activate the smart cap so that the loaded API can be released. The flexibility of the system allows different APIs to be loaded, such as chemotherapies, immune modulating drugs, siRNAs, and isotopes. The synergism between mesoporous nanoparticles and DNA molecular machines is created by the combination of the acquired advantages per each discipline. For example, nanoparticles benefit from the EPR effect. EPR stands for endothelial permeation retention effect, which is considered passive targeting. Because the nanoparticles are covered by DNA, the cancer cells actually actively endocytose the particles as a Trojan horse, outside looking like a DNA chunk, but from the inside, including chemotherapy. The smart caps allow specific unloading of the API, and a double peak effect is seen in the release profile of the API. First peak happens in 2 hours, and the second after 1 week. The above characteristics in turn allow substantial advantages over current treatments such as increased efficacy and decreased toxicity. The idea being that a higher percentage of the API reaches its target cells. TXN 770 is made from a silica core loaded with doxorubicin and capped with a DNA enzyme, which responds to stimuli of ATP plus magnesium. ATP is more prevalent in its free and magnesium-bound form in cancer cells compared to the normal cells. Magnesium role in cancer exacerbation, including in cancer migration, metastasis, cancer stem cells, and cancer resistance to anthracycline, has recently been elucidated. TXN 613 is made from a silica core loaded with mitoxantrone. It is capped by gossypol, a second chemotherapy that synergizes the activity of mitoxantrone. TXN 613 responds to low pH levels below 6 plus high lactic acid levels. The low pH is characteristics of cancer cell and especially metastasis. Cancer cells secrete lactic acid to their environment to cloak themselves from the immune system. TXN 613 is covered by an active targeting agent. TXN 252 is made from a silica core loaded with docetaxel. The cap opens at high levels of an enzyme which is abundant in cancer cells, plus low pH. Docetaxel has a 200 times lower IC50 compared to doxorubicin. TNBC, or triple-negative breast cancer, is an aggressive form of breast cancer presented in younger women with a more advanced stage of disease. No effective targeted treatment is currently approved for triple-negative breast cancer. As its name implies, it is negative for all three receptors of Herceptin, estrogen, and progesterone. Regulatory agencies recently published substantial incentives to advance the development of triple-negative breast cancer treatment. Triarch's Nano will benefit from these incentives, such as PCR guidelines allowing approval after phase 2 using surrogate endpoint of PCR pathologic complete response instead of survival data. 505(b)(2) process allowing only bridging studies to show as safe and as efficient results, and orphan, accelerated, breakthrough, and fast-track status. As a reminder, this is the structure of TXN 770. The logic behind the specific programming of our nanoparticle TXN 770. Magnesium role in cancer development is elucidated more and more as new papers are published. The MMM model, the relation to mTOR, PI3K, AKT are all now well recognized. Lately, magnesium's role in creating the resistance to doxorubicin in triple-negative breast cancer cells was elucidated. Magnesium was found to modulate doxorubicin exit from the cells through lysosomal sequestration. These graphs show the unloading of doxorubicin dependent on the levels of magnesium and ATP. Below, the specific cytotoxic effect of TXN 770 in triple-negative breast cancer compared to normal breast cancer cells can be seen. The study below shows the comparison of TXN's cytotoxic effects between cancer and normal breast and hepatic cells. Another study was done showing the cytotoxic effect is not due to the silica or the DNA enzyme, but the doxorubicin that is unloaded from TXN 770. We explored the mechanism by which the nanoparticle enters the cancer cell and found it was related to PI3K and early endosomes. We compared TXN 770 to Doxil, liposomal doxorubicin, both in normal and triple-negative breast cancer cells, MDA231. As can be seen, we had 50 times greater cytotoxic effect compared to Doxil and roughly 3 times better safety. As can be seen from the graph, circled in red, showing the quantitative comparison. In the picture below, the qualitative comparison, where blue are the triple-negative breast cancer cells and red is the doxorubicin released from TXN 770 compared to Doxil. Following the encouraging in vitro results, we followed with in vivo studies. These studies showed greater efficacy and safety compared to free and liposomal doxorubicin. Intratumoral study showed smaller tumor sizes and better survival. Histology of the slides supports the measured animal results. The histology shows gamma A2X DNA intercalation, the mechanism by which doxorubicin works. In the first IV study, we show better results in the treatment group. Again, histology supporting the animal results. Intraperitoneal study was also done, showing better results in terms of histology and safety in the animals. A second IV study was done comparing head-to-head the effect of Doxil, liposomal doxorubicin, an approved product with sales in the billions of US dollars compared to TXN 770. We used 4 times more Doxil compared to our treatment. Again, only 50 micrograms of TXN 770 were used comparing to 200 micrograms of Doxil. The results were that in terms of the tumor size, the mice weight, and the histology, TXN 770 at 25% of the dose had much better results. In terms of histology, as can be seen in the graphs and the slides, we had 5 times better gamma H2AX coloring compared to Doxil. Another interesting result was the penetration of TXN 770 particles deep inside the tumor bed. We believe this is due to the phenomena we call the Trojan horse effect. The DNA coating the particle causes the cancer cell to uptake the particles as they think these are chunks of DNA. The second particle we developed is called TXN 613. It is loaded with mitoxantrone, a second-generation anthracycline capped by a gossypol capped, an additional chemotherapy for a combo treatment. The particle is covered by an active targeting molecule. Its caps relates to low pH below 6 and high lactic acid levels, which are known to be abundant in cancer cell metastasis. As can be seen from the graphs on the left, the release of the API is very accurate, having almost no release in normal pH levels and normal lactic acid levels. In vitro studies shown compare the normal and triple-negative breast cancer cells using each of the chemotherapies alone, mitoxantrone and gossypol, and in combination showing the synergistic effect. In vitro tests using the most aggressive PDX models we could locate, being poorly differentiated adenocarcinoma, metastasis from the liver which recurred or was resistant to all chemotherapies commonly used, including docetaxel and liposomal doxorubicin, has responded to our treatment. As can be seen in the graphs, the blue line shows the full treatment dose, the purple line shows the half dose of the treatment compared to the placebo size. In the red line, the free chemotherapy mitoxantrone was given, and all the mice in this group had died due to weight loss. As can be seen, the mice receiving TXN 613 all gained weight, whilst the one receiving the free chemotherapy lost weight and died. To conclude this presentation, I would like to summarize the information shown. Triox Nano's patent depicts the combination of DNA machines and porous substrates to create an intelligent delivery platform we call SMART. To date, 3 particles were discovered, each having unique features to deliver doxorubicin, mitoxantrone, and docetaxel to triple-negative breast cancer. While the company focuses its effort on advancing its triple-negative breast cancer solution, we welcome cooperation with strategic partners interested in developing other particles using our platform technology. For more information, please visit our website at www.trioxnano.com and look at our recently published papers.
Thanks, Roy, and to everyone for their excellent presentations. Now it's time to move on to the roundtable discussion with the speakers. We've heard about a number of very different types of delivery technologies today and about some of the therapeutic areas they're currently being applied to. So now I'd like to get your views on how you would like your technology to be used in future applications. If we could please start with the thoughts of Paul at Phosphogenics.
Thanks, Ravina. Look, we see our TPM being a great addition to the general formulation toolbox to overcome a range of formulation and delivery challenges. So in the coming years, we would obviously like to see TPM used in a range of commercial products with a range of partners and across a range of different routes of administration. And we see it providing technical and therefore commercial differentiation. Now, I guess one of the differentiation aspects that we're most keen on is patient safety. And so in the context of injectables, you heard during my presentation that I mentioned Cremophor, which is an excipient used to dissolve drugs with very poor aqueous solubility. Now, Cremophor is basically castor oil, which is obviously not ideal to inject into your veins. And it comes with a range of toxic side effects that make it unusable for certain patient populations. So look, we've already conducted studies demonstrating that we can dissolve these drugs using TPM in the absence of Cremophor, and the ability to replace excipients like this with basically vitamin E would be a much safer option for patients. And the same, I guess, is true of our opioid patches, where safety is our main priority. So, I mean, we're trying to deliver opioids in a profile that successfully manages pain, but also limits the adverse side effects, which are a burden for that particular therapy. Same with accidental overdose, that's much rarer in a transdermal patch, and it's also a less favored dosage form for abuse. So, I mean, if I come back to the original question, I mean, one of the things we'd love to see our TPM doing is being instrumental in the creation of safer drug products.
Excellent. Thanks for that, Paul. And now if we could move on to hear the thoughts of Kevin at TissueGen.
Well, thank you. Good question. I believe our technology is really ideally suited for 2 different major application areas. First is enabling innovation in current medical devices. For example, we could use our technology to load antibiotics or other drugs into a standard hernia mesh. The other area where I think we can make a major step forward is in tissue engineering and regenerative medicine. I think here we can really maybe potentially change current paradigms. You know, if you think about the spinal cord regeneration work that I mentioned in our presentation, by applying concentration gradients to these chemotactic agents, we can purposefully guide tissue during wound healing in the central nervous system. That potentially could make a major step forward in treating spinal cord injuries. There's other applications, for example, that it'd be fascinating to see how do in vitro studies to see how cells respond to these concentration gradients. I think creating tissue in vitro is a reality with this technology. And as the technology grows and matures and begins to move into the clinic, I think we could ask very fascinating questions. You know, for example, if we can recruit cell types, could we induce endothelial cells, vascular endothelial cells, to migrate into a small diameter graft? I think it'll be fascinating to see where our customers will deploy and innovations they will make within the body.
Brilliant. Thanks for that, Kevin. And now if we could hear the thoughts of Patrick at Portal Instruments, which other applications would you like your technology to be applied to?
Thank you. So for us, our mission is really to remove needles from drug delivery. And really the overall goal by doing that is to really improve the patient experience. So I think we won't rest until we've removed needles from current drug delivery. And for us, the immediate goal is to get this device on the market such that it can be used by patients, so we can really learn its benefits and perhaps also its shortcomings. Then, as we evolve the technology, we see it sort of addressing other unmet need in very hard to deliver areas. So, for example, delivering to the eye is very challenging and can cause great anxiety for patients if they have to get a needle inserted into their eye on a regular basis. Conversely, there might be great benefits to inject directly into the tympanic membrane or to inject directly into joints. So that's how you'll see our technology evolve over time. And however, for now, we're very, very focused on addressing the unmet need for the injection of high viscosity biologics.
Excellent. Thanks for that insight, Patrick. And now if we can move on to Roy at Triox Nano.
Yes, thanks, Ravina. So I expect Triox Nano's smart platform technology to be used in vast fields of pharmaceutical, veterinary, botanical applications. The system is very flexible, and the synergism from combining the DNA molecular machines and the mesoporous nanoparticle is huge. On one hand, we can carry different payloads, such as chemotherapy, antibiotics, siRNAs, isotopes, and so on. On the other hand, our smart caps can recognize a combination of different variables and exert their loading or unloading function based on a pre-programmed basis. So, as an example, in the literature, we know that breast cells have high magnesium levels, so we created a DNAsome that relates to magnesium and ATP. On the other hand, prostate cancer cells have high zinc levels. We developed a cap that relates to zinc and ATP. So Triox Nano is focusing on triple-negative breast cancer, but we are looking to collaborate with other groups on other oncologic indications. We're working on ovary, pancreas, and prostate cancer. And we're also cooperating with other companies on treatments for MS, inflammatory disease. And I believe the future of drug delivery platforms are biologic computer-based nanomachines. Thank you.
Excellent. Thanks for that, Roy. It's good to hear what you're up to. And finally, if we could hear from Joe at Corium, what do you think about any potential future applications?
Thanks, Ravina. So, historically, I think people have really looked at alternative methods of drug delivery as a life cycle management tool. But we really think that there's an opportunity to, use alternative drug delivery options to enable new drug candidates, especially for relatively poorly soluble molecules, which are particularly good candidates for our, Corplex transdermal technologies. And in that context, we're, typically able to do a quick paper feasibility within a couple of weeks, and then develop a prototype formulation to evaluate the feasibility of transdermal delivery within a few months. So it's relatively easy to incorporate transdermal delivery within the current drug development process. And as Paul mentioned, we think that there's really opportunities for dramatically improving the safety profile of drug candidates. By avoiding the GI tract, you're able to eliminate or reduce GI side effects, avoid the first pass effect. And for drugs that have problematic Cmax-related side effects, by delivering more sustained and controlled fashion, we can also improve the safety profiles of those agents and keep drugs moving forward that might otherwise be viewed as unsafe. And I think finally, improving adherence, particularly for chronic conditions, is a ubiquitous problem for the industry. And we're really focused on developing multiple day, ideally once weekly transdermal patches that we think provide a simple, convenient, and inexpensive way to significantly improve patient adherence and the overall patient experience with new drugs. Thank you.
Thank you. Thanks for that, Joe. Okay, great. I think we've got time for just one more question before the end of the session. And now I'd like to look at the whole drug delivery market as a whole and what everyone sees for the future. What do we think in terms of therapy types of the new innovations that might need new delivery methods to enable them to be delivered in the future? And if we could start with Patrick, please, at Portal Instruments.
Yeah, I think I've been really astonished by the diversity of technologies that we've heard about this morning. And I think that really highlights how drug delivery really addresses unmet needs and give, in particular, the pharmaceutical industry and the pharmaceutical companies additional edge in the marketplace. And I think we'll continue to see that. I thought, Sanofi had this great motto a few years back, which was beyond the pill. And I think that's where we see the whole field of drug delivery evolving. It's going even beyond just differentiation or giving the additional edge into the market. It's also, as we've just heard from the last speaker, it's perhaps having an impact on adherence, on compliance, and in the end, improving outcomes for patients. We see from our side a huge opportunity around digital health, how you can basically weave the device and the therapy into the patient's life such that it becomes a habit, such that it becomes something that they do naturally and so on. So that's how we see the evolving field of drug delivery.
Brilliant. Thanks for that, Patrick. And now, Joe at Corium, what do you think about the future of drug delivery?
Sure. So, we think, one of the key macro trends in the pharmaceutical industry is really a move towards customized medicine and customized solutions for patients. And that conversation tends to really be framed in terms of tailoring the particular therapy to the biology of the patient. But I think another way to think about that is also to think about, what delivery methodology is appropriate for particular classes of patients and or types of diseases. And particularly when you're thinking about chronic conditions in CNS and neuropsychiatric conditions, where the nature of the disease in and of itself makes, adherence and compliance problematic. Again, we think, going to non-oral options for some of those patients, particularly for, transdermal patches that can decrease the frequency with which patients need to take their medications and can deliver, that chronic medication at a consistent level and avoid, the peaks and troughs and other issues associated with that, we think is an important factor. And it's well known that there are, a subset of patients that really do prefer to avoid oral therapy, both from compliance perspectives and also, swallowing issues, et cetera. So we think thinking about that and thinking about how you really get the patient to have the best overall experience and improve compliance is really an important thing.
Definitely. Thanks for that, Joe. And now if we could move on to Roy.
Thank you, Ravina. So again, I believe the future is the biologic computer-based nanomachines, because what we showed in certain in vitro studies is that we are able with these machines to concentrate the API at the target cell. And I think that's the main issue, avoiding the high toxicity and allowing high efficacy. In a head-to-head study that we did versus Doxil, we saw 20 times higher doses in the cancer cell delivered by our system. So I really believe that if we're looking not for the near 5 or 10 years, but for the far future, this would be the basis for treatment.
Excellent. Thanks for that, Roy. And now if we could move on to Paul, what do you see for the future of the industry?
Look, I guess putting my old formulators hat on, I think one of the things that the actual drug delivery market's going to have to do is move in response with the rise of biologicals. So, I mean, they're horrible to formulate. They're large, complicated molecules. They've got tertiary structures that you have to maintain for drug activity. And really, their routes of administration currently are limited to injection. So you've heard Patrick already look at overcoming this, but moving forward, I think administration routes are going to have to adapt. And I know people are already looking at oral delivery technologies and other things that can deliver macromolecules, because as we've heard already, I mean, people would rather prefer to avoid injecting these drugs all the time.
Great, thanks for that, Paul. And now if we could finally finish up with Kevin, what are your thoughts on this topic?
Well, I agree with what has been said. I think biologics are key to this. Today we've heard lots of very innovative advances. It's been a very interesting webinar. Thank you. As far as therapies, I think we still have fundamental questions that still need to be answered, though. We need to figure out how do we actually get the drug to the target, for example, remediating solid tumors, crossing the blood-brain barrier, the retina blood barrier, remain formidable challenges, and I think conquering some of those basics of the biology is still another area that's ahead of us. Thank you.
Excellent, thank you for that. Well, I'm afraid that's all we have time for today. If you have submitted a question that hasn't been answered, we will follow up via email. I would like to take this opportunity to thank Dr. Patrick Anquetil, Dr. Joe Sarrett, Dr. Kevin Nelson, Dr. Roy Fafara, and Dr. Paul Gavin for their informative presentations and for answering our questions today. And of course you, the audience, for watching. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again for our next webcast.